Hello guys, I am Shubham from ShubhVed Tutorials. This video is about unordered tetrad analysis. So basically what is unordered tetrad? That is a random arrangement of spores, right, inside the ascus. As you can see this in the Saccharomyces cerevisiae, right? This is the yeast basically. And now we will see how to do analysis of using unordered tetrad. First of all, this unordered tetrad analysis is used in dihybrid crosses. Okay, two genes basically, right? Two genes that are linked or assort independently, right? So this is used for dihybrid cross, or we can say two genes that those two genes are linked or assort independently, right? Okay, so in this case we use unordered tetrad analysis. So these are the two chromosomes, and capital AB are the genes, and there is you part small a small b. Okay, first we will see here no crossover. No crossover means as it is means capital AB and small ab as it is. Now the second, if double crossover happens between the two strands, okay, double crossover two strands, then what we will see? Okay, so these are the chromosome. I will draw here for simplification only those parts, right? And cross occurs between these two, double crossover. Okay, so see the path here now. This down again this. So we can see same AB here also AB. Then this, right, means it will come again down only. So we will see the same types, right? So double crossover also two strands we will see. Okay, so this is known as parental ditypes, right? When there is no crossover and when double crossover occur between two strands, we see parental, same parental combinations are seen. So this is known as parental ditypes. Parental ditypes. Okay, ditype means dominant and recessive. Okay, so these two combinations we can see when there is no crossover and when the double crossover happens. Now, double crossover involving four strands. Okay, just I will draw your strands only. I will draw strands only. Okay, these are the four strands. Then the first crossover between these two, and second crossover between this. Okay, we know here is A, here is B, right? Again, small a, small b. Okay, so this is the cross. See carefully. So first strand starting from here, okay, goes down, right? So first strand AB. I will just write, okay. Then second strand, second strand. Starting from here, goes down in here. So this is also capital A, small b. Right? Now third strand. From here, goes up. Okay, and small a, capital B. Again, starting from here, goes up. Okay. So here we can see all are recombinants. Okay, no parental ditypes. So these are known as non-parental ditypes. So this is the case when double cross over involving four strands. Okay, all strands are involved. Right? Now double strands involving three strands. Okay. Again, these are the chromosomes. Right? Now the crosses between three strands means first cross here and second cross here. Okay. Then we will see now what will happen. Here are AB genes. Okay, so first one will come as it is because there is no cross involved. Right? Now second one, this down again, this whole capital A, small b. Right? Now here, starting from here, goes up again here and down. Okay, down means what? Small a, small b. Okay, down strand, right? Here it is b. So from here it goes up and down, right? So small ab. Then next strand here goes up and so a b. Okay, this is double crossover involving three strands. Double crossover, three strands. Right? So here we can see half parental, half non-parental, right? 50-50%. Also, when there is a single crossover between two strands, single crossover, we'll see what is single crossover. Again, I will draw your chromosomes. These are the chromosomes, right? Now, and here are the genes. Capital AB and small ab. So cross is between this two. Single crossover. Okay. See, up, same. Right? This will be changed. How? Like this. Right? Again. Okay. So here again will be, and this will be as it is. So in this both of the cases, we can see what we can see here, parental as well as non-parental ditypes. Right? So this is also known as tetra type. Tetra type. Okay, tetra type means it is also known as TT. TT. Okay, tetra type. So here we can see parental and non-parental types. Both of 50-50% we can see. Right. So this is a this we have to remember that when we will see parental ditypes, when there is no crossover. Okay, so we will see a quick summary here. When there is no crossover, right? So when there is no crossover and when there is double crossover, we can see parental ditypes, right? Then when the double crossover is involving four strands, Then we will see all non-parental ditypes. Okay, non-parental ditypes. NPD, this is known as NPD. NPD, non-parental ditypes. And when there is double crossover involving 3 strands and single crossover, okay, between 2 strands, so there will come parental as well as non-parental ditypes. Means we will see 50% parental combination and 50% recombinant combination. So this is known as tetra type, that we'll see in this case. Okay, so now there is a formula to for the analysis of unordered tetrad. Okay, so the formula is NPD, that is non-parental ditype, plus half into tetra type upon total number of tetrads into 100. Okay, this is a formula to find the map distance using unordered tetrad. Okay, so that's it for this video. I hope this video helps you. If this video helps you, do let us know in the comment section. Thank you guys for watching this video. If you like it, please share it. Don't forget to subscribe and press the bell icon to get notified whenever new video is out. Also follow us on all our social media pages, and you can join us on Telegram and stay connected for the latest updates. All links mentioned in the description section.